rows that are missing like 5 in, and then
So from most of my education is in Catholic, and most of it was Jesuits, so pretty progressive. So it has many issues specific.
Hope you didn't sit at the same time.
Yeah. I think it's more of a Christian thing.
It's not a scientific thing. Alright. Now we have fun.
I think we're doing stress now.
Stress.
We're doing stress?
This is my idea. I can do this for advice.
Are you feeling
I don't know what I'm going to talk about. All right, let's get started. First of all, number one and most important, not this Sunday, but the Sunday after is the last day before the second midterm, and I'll have a review session again. So I've reserved space. It is going to be, oh, come on, from 2 o'clock and Anderson 112. So that's the Nelson Auditorium where the seminars, Friday seminars are usually. Okay? I couldn't get that one, I couldn't get this one, but we got the one downstairs. So 2 o'clock on Sunday. It's the Nelson, this one right down there. So it's first floor. As you're coming in the front, it's the far left. All right, so from now until the midterm, we're going to be talking about my favorite topic, the topic I've spent probably my entire career studying. So, stress. What is stress? Somebody define it for me. None of you have ever felt stress ever before. Yes?
From a physical standpoint, it's a change in homeostatic balance that needs to kind of be reset back into our homeostatic balance.
Okay, so change in homeostatic balance. So, great idea, but tell me, one of the biggest ways in we can disturb our homeostatic balance is by consuming a meal. You guys are going to really, really, really upset your homeostatic balance on Thursday. Guaranteed. Okay? Is that stress? And if you say yes when I overeat, okay, but what makes the difference between regular eating and overeating? Yeah?
There's a range involved that you have to go past that ends up making it chronic or acute, where it actually ends up throwing off.
Hold on, wait a second. You just threw out two completely new words, acute and chronic. What are those?
So you could have stress that ends up peaking for like a short period of time. You can have stress that ends up continuing and actually eventually causing issues with that throughout your body. And it's not just like a certain there's like a set point, but there's a range where your homeostatic things could be between.
I'm still trying to figure out, you just drew a line. You called it a threshold. I have absolutely no idea where to draw that threshold. Where do we put it?
Person dependent.
But how? How do we define this? I mean, we have to be able to figure out what it is to study.
Your cortisol levels.
Well, okay. Cortisol levels. People tell us that cortisol is a stress hormone. How do we know that? Cortisol goes up with stress, right? What is stress? How do we know that it goes up with stress if we haven't yet defined stress? Yeah.
It'd be like preparing your body for an unpredictable event that's challenging.
Okay, now we're going to talk about preparing the body. That is completely different than a disturbance in homeostasis. Because preparing automatically tells us that you haven't yet had a change in homeostasis. So are we throwing out the whole homeostasis idea entirely? Yeah?
I mean, it's preparing usually in response to some stimulus. So,
What stimulus? So preparing to eat? I mean, if we can talk about it, let's come back to homeostasis. Okay, we're going to prepare ourselves for a change in homeostasis. That's going to be stress. Well, we know we're going to change it 3 times a day. If we're lucky, we're going to change our homeostasis by eating 3 meals, right? Are we preparing for that change in homeostasis? Is that preparation stress? Yeah?
Could, like, the preparation process itself disturb homeostasis? Like, if you're working yourself, like, making an exercise? I'm not sure. Like, if you didn't have.
Well, now we just brought in another concept. Are we using too much energy? How much energy do we need to know? If we go out and take a run, take a jog out there, we're expending energy. Is that stress? Yeah?
Sustained mental or physical exertion.
Sustained mental or physical exertion. Maybe. But people run long distances for fun. You sit down and you read a book, spend 3 or 4 hours reading a great book. That's sustained intellectual activity, right? Is that stressful? No, it's pleasurable, right? So it's not. This is exactly the opposite. Yeah.
Could it be in response to noxious stimuli?
Okay, great. It's a response to noxious stimuli. What is a noxious stimulus?
Something that's harmful or unpleasant?
Harmful, unpleasant. What is unpleasant? Yeah? So maybe anxiety. How do we know we're suffering from anxiety? Yeah?
You look for changes in behavior.
What changes behavior? I walked in this classroom, started lecturing. I changed my behavior. Is that stress? Facing all you guys every morning? Yeah. Yeah.
Maybe from the perception of threat, or what we perceive to be a threat.
Okay. What's a threat?
Something that can pose harm to your physical well-being.
Okay.
Necessarily.
A lot of people tell me they have stress from taking exams. That's not going to hurt your body. Might hurt your mental, but what?
But your brain might not know that.
Okay. So maybe we have to think about the brain. Come on, isn't this an easy question? Yeah.
a continual preoccupation with something. Like, I feel like if you're stressed, you're continually thinking about it and expending energy on it.
Maybe. But I'm just thinking again, if I'm reading a really good book and have to put it down to do something, I'm thinking about that book, thinking about the story, and if it's a big book, I'll probably be thinking about it for several weeks. I wouldn't exactly call that stress, but I'm preoccupied with it. Yeah?
It's like the process of like perceiving and not just stimuli as threatening or challenging, and then the reaction to it in preparation.
Yeah. I mean, it's an interesting idea, but again, I can think of lots of examples in which what you're describing is not actually stressful. Before we go into this too deeply, I just want to point out, I did a search a number of years ago on a Google search on the word stress. And it came up with more hits than heart disease, than obesity, than mental illness, than heart attacks or cardiac disease. The only word I could find, stress-related word, that had more hits than stress was cancer. And cancer has lots of different diseases. And so this is an incredibly important, well-used word. And probably every single one of you walked into the room today saying, I know what stress is, and yet we just spent 5 minutes and nobody can define it. Think about that for a second. Okay? This is my favorite definition of stress. Okay, does that sum up what you guys think of as stress? Now here's the problem. Look at this. Feeling, desire, deserving. Quantify desire for me. Quantify feelings for me. Here's a fundamental issue in science. And if you probably take away one thing from this class, other than its receptors, is that if you can't measure it, you can't use science to study it. Science is a way of quantifying and measuring things. And in fact, I work up in the far north in Alaska for many years, and I was told that the Inupiat, who live up there, their word for scientists, when translated literally, means one who measures things. And I love that because that's essentially what we as scientists do. We measure things. If we can't measure it, we can't study it. We can't use our statistics to look at it. We can't do anything else. So the study of feelings, the study of desire, the study of deserving, those are the humanities. They're not sciences. Okay? So if we want to study stress in biology, we've got to figure out what we're going to measure. Okay? And that's the key thing. That's what we're going to start talking about today. All right. So, if we're thinking about stress, this is the traditional model. By the way, I should point out that scientists haven't even come up with a good definition of stress. This is still one of the open aspects of this, and about every 5 to 8 years, somebody puts in press that we should stop using stress entirely, and this is a scientific word, because it's just meaningless. There's no way we can define it, and if we can't define it, we can't measure it. If we can't measure it, we can't study it. But this is what people have started to talk about. Is that an animal is in a state of dynamic equilibrium. So this comes back to the homeostasis suggestion. This is what the animal is in. State of a dynamic equilibrium. It's not equilibrium, because things change. You eat, you got to come back. So it's dynamic equilibrium. Okay? And then something happens to disturb that dynamic equilibrium. It's some sort of a noxious stressor. Somebody over here talked about noxious. Again, we have to figure out how to define noxious, but it's a noxious stimulus. And this is what is generally called a stressor. Stressor, something that causes stress. One of the things that we have to talk about is that the word stress has come to mean 2 and maybe even 3 entirely different concepts. Okay? Something stresses us out. Well, that's something people call stress. But that's really a different concept than the change that occurs in our bodies in response to that stimulus. Right? 2 entirely different things. So we call this, the noxious stimulus, a stressor. And that disturbs our dynamic equilibrium. And if this animal does not solve that problem, it is in deep trouble. Probably dead. So what happens is that there is an adaptive response, which people have called the stress response, and that serves to reestablish that dynamic equilibrium. So we have stressors and we have stress responses. However, and here's the other key concept here, which is that is probably not what any of you are thinking of when you talk about suffering from stress or being in too much stress. What you're probably describing is this situation, where the adaptive response was supposed to be the adaptive response. The stress response overcompensates for your noxious stimulus and serves to disrupt that dynamic equilibrium itself. This is what we call stress-induced disease. So this is why I talk about this. 3 different concepts here that people use with the word stress. The nature of the stimulus, the nature of the response, and the determination that the response actually is causing the problem itself. It's not actually adaptive, it's maladaptive. So traditionally, this aspect has been studied by the psychologists. What causes stress? What are the stressors that are taking place? And this aspect of it traditionally has been the realm of biomedicine. Why is stress causing disease? This response has generally been understudied, and for many, many years, most physicians and psychologists were mostly uninterested in how this actually took place, mostly because the first person who took an attempt at this turned out to be horribly, horribly wrong. Okay? And he was so spectacularly wrong that for about 50 years nobody else attempted to try to figure out how this balances itself. This has basically been what I've been studying, how this works. But there are 3 different concepts here. Okay? Yeah.
The first adaptive response where we restore dynamic equilibrium, isn't that kind of like any other stimulus that you talked about, like eating or running? Because we're going back to that.
So the idea is this dynamic equilibrium takes care of things like eating, circadian rhythms, things like that. This is a noxious stimulus. And we're going to be talking about what actually is a noxious stimulus, hopefully later today. Okay? All right. So key to all of this is this. You have this stressor. This poor bird is about to get eaten. Okay? This is a major stressor. I think we can all agree that this eagle is a stressor for that bird. Therefore, in order for this bird to survive, not only does it have to escape, but this process of detecting and coping with this stressor has got to lead to survival. And that's what this system was designed to do. And there are essentially 2 major arms of this response. And this figure may be the most important figure for the next 2 lectures. And we're going to go into a lot of detail about what is taking place inside of all these boxes. But essentially, the brain is detecting the stressor, and we're sending us 2 different signals from the hypothalamus. But one of them that we've talked about is going to the pituitary through the HPA axis to the adrenal cortex, where we're releasing the glucocorticoids from the adrenal cortex. This is a fairly slow response. The first response, however, is a very fast response. And this is the activation of the sympathetic nervous system, which then sends a signal to the adrenal medulla. So not the adrenal cortex, but the adrenal medulla, where we're getting the release of epinephrine and norepinephrine. And the epinephrine and norepinephrine have a variety of different effects on the body. So for the first little bit here, I want to talk about this response. It's so fast, and the catecholamines activate what we have come to call the fight or flight response. Okay? Yeah.
I've also heard that people say fight, flight, or freeze. I don't know if you have any opinion on that.
Yeah, the fight or flight is simply, in many ways, a shorthand. Yeah, there's fight, flight, freeze. A lot of animals freeze. A lot of times it's different. Boy, there's this absolutely amazing ecology paper that I read a number of years ago. So if you have a rabbit that's being chased by a predator, and you'd think that it would want to run away. In about 10 to 20% of the times, the rabbit actually runs towards the predator. And what it does is it actually is just as likely to escape if it runs towards the predator than if it runs away. And what it does is it shortens the time span that the predator has to attack. And so by going straight at the predator, it sort of freaks the predator out, and decreases its reaction time, and the rabbit actually has just as big a chance of escaping. So it's not just fight or flight. I mean, it's not going at the predator to attack it. It's not fight, right? But it's also not fleeing. So it's, all the fight or flight really is a shorthand for these catecholamine responses. Okay? All right. We'll come back to this, but let's think about this catecholamine response. Where is it coming from? So this is a little bit of review from last week. Here's the adrenal glands right on top of the kidneys. And here are the two different layers of the cortex and the medulla. So the medulla's in the middle, and here's the cortex. Now remember, the blood is flowing this way, so the last thing that it hits is the medulla. And this is where all of the epinephrine and norepinephrine is coming from, the catecholamines. What do the catecholamines look like? So they start off, the production comes from phenylalanine. It's an amino acid. It's converted into tyrosine, and then there's a key enzyme, tyrosine hydroxylase, which converts it into dihydroxyphenylalanine, or DOPA. And then DOPA is then converted by DOPA decarboxylase into dopamine. And that is a very important neurotransmitter in the brain. But dopamine is then converted through dopamine beta hydroxylase into norepinephrine, and then this from phenylethanolamine N-methyltransferase, or as I always call it, PNMT, and this is the rate limiting step. And that then converts norepinephrine into epinephrine. And epinephrine is one of the most important and a major catecholamine in the body. Norepinephrine is also, but epinephrine is much more potent in effect. Yeah.
So this is all happening in medulla?
Yep, inside the cells. This actually happens all throughout the brain too. Both epinephrine and norepinephrine and dopamine are major neurotransmitters in the brain. But in terms of the endocrine function, this is happening in the medulla. Okay? So here's some examples of the catecholamines. So right up here on top, here's our epinephrine, and here's our norepinephrine. By the way, does anybody know what the difference is between epinephrine and adrenaline? Yeah?
Brand name?
Not quite, but close.
It's a patent, right?
I don't think it's a patent, but adrenaline is the name for this molecule in Europe. And epinephrine is the name for this molecule in the United States. So, yeah, an adrenaline rush, it's the same thing as an epinephrine rush. So when people are taking epinephrine, they're taking adrenaline. So now you can start to understand why epinephrine is banned by a lot of sporting groups. Another thing, here's ephedrine. So ephedrine, pseudoephedrine. These are the things that are present in Sudafed and a lot of the cold medicines. It's why the Olympics are always worried about who's taking Sudafed before having competition. It's because they're basically hitting themselves up with epinephrine. Here's another one. Here's amphetamine. So the amphetamines are very much catecholamines. So they're having much the same effect as adrenaline or epinephrine. On the flip side, this is propranolol. This is an antagonist for epinephrine.
Isn't that a beta blocker?
Right. So we're going to talk about this in a second. There's two receptors for epinephrine. There's the alpha receptor and the beta receptor. As we're going to find out in a second, the beta receptor is the one that produces most of the effects of catecholamine. Propranolol is an antagonist for the beta receptor, which is why it's then called a beta blocker. Again, another drug that is heavily used in performance. So I have seen estimates that as many as 2/3 to 3/4 of all concert musicians take propranolol. They take a beta blocker before a concert. Why? Try to get mellow. so that they're actually playing and not allowing their nerves, their anxiety, their stress response to interfere with their playing. It's another big number of sports that actually also try to abuse beta blockers. Most of the shooting sports, for example, where you actually have to shoot a target, especially when you're far away, a heavy breathing rate will actually sit there and move your gun. So in the Olympics, a lot of those shooters will try to do something to decrease, and beta blockers was a major problem many years ago. Okay, so all of this starts to make maybe a little more sense when you start seeing that all of these compounds are essentially mimics or inhibitors of your fight or flight response. Okay, so how do these things get released? I've always thought this was really cool. So let me back up a little bit and talk about what the sympathetic nervous system. So the sympathetic nervous system, we have a signal coming down through the spinal cord, which then sends an axon to the ganglion. And then it so what it's doing is there's a ganglion here, and the axon comes down, innervates the neuron in the ganglion, and releases acetylcholine, or ACH. This neuron then sends its axon down to the muscle and releases usually norepinephrine or epinephrine right onto the muscle, and that's how you're getting the sympathetic activity in the muscle. So the classic sympathetic activity is this pathway of going from the spinal cord to the ganglion, then from the ganglion neuron sending the signal to the muscle. And these ganglia are all up and down the spinal cord, forming this big line of ganglia. So that's the sympathetic nervous system. For the medulla, we have something a little different. It is leaving the spinal cord and going through the ganglion. It doesn't stop. The axon goes right through the ganglion and ends up in the adrenal medulla. So here's the whole system in much bigger detail. So the cell is in the spinal cord, sends its axon out through the root of the spinal cord, and instead of stopping, goes through it. So its axon passes through the ganglion, and then comes down into the adrenal medulla, and this is where the synapse occurs, and this is where it's releasing its acetylcholine. And then the acetylcholine is activating a chromaffin cell. And remember, we talked about a chromaffin cell is from the chromaffin dye that's binding to, it turns out, catecholamines. So epinephrine and norepinephrine. And by doing that, it stimulates this cell to release epinephrine and norepinephrine. It's the same thing as this neuron that released the norepinephrine onto the muscle, except that this cell has no axon. And so instead of sending its axon down, it just releases it into the blood. And so you get the release of the norepinephrine and the epinephrine into the bloodstream. So what the adrenal medulla really is, it's essentially a modified sympathetic ganglion. So it is essentially like this ganglion here, except it's in the adrenal medulla and has no axon. This should automatically be telling you why the fight or flight response is so fast. Because it is essentially neural, not endocrine, until the very end. All right? Yeah.
What about when athletes look at reaction times to like different, like maybe like passing the ball?
So the reaction times are different. That's directly neural onto the muscles. So that's not a fight or flight response. Okay? You do not have to have a fight or flight response in order to be active. So people who are running, things like that, they've actually done studies on horses where they put horses on treadmills and watch the horses run. They do not have a stress response. Humans do not have a stress response just because we're active. Okay? It's the fight or flight response occurs because you are facing some noxious stimulus, not something like a sporting event. Now, if it's a championship and you get really nervous, then you're having a fight or flight response. All right, any other questions? All right. So let's look at the whole thing inside the medulla in more detail. So here is the inside of the medulla. These are our chromaffin cells. So they're the ones that are storing and packing the epinephrine. Here's our axon coming down from the spinal cord, and here's our axon terminal. So there, the axon is then going to be abutting the chromaffin cell. So if we take this and blow it up into even more detail, this is what we're looking at. Here's our axon terminal, here's the acetylcholine flowing out into the synapse and into the synaptic cleft. As the acetylcholine binds to its receptors, we're going to get the influx of calcium. And that calcium is then going to stimulate the exocytosis of our vesicles that are containing epinephrine and some dopamine as well. But the epinephrine and norepinephrine dumped right into the blood flow that's coming down. And that's how we get the release. One other important thing here, and that are the glucocorticoids. They're also, oh no.
Are you having a stress response right
I'm not having yes. That did not seem good. Because I didn't do anything. Yay! Okay. No problem. Glucocorticoids. Glucocorticoids are a big part of this process. Now remember, the glucocorticoids are coming down from the other part of the adrenal, from the cortex. And so they're passing right by all these cells. And what they are doing is stimulating the chromaffin cells to produce more epinephrine. So, we have norepinephrine being converted into epinephrine. The enzyme that does that is PNMT. The glucocorticoids are increasing the production of PNMT. So this is a classic kind of effect. Permissive, thank you. Now I'm thrown off my game. All right, so this is a classic permissive effect. The glucocorticoid. So they are really important in stimulating the increase in the production of epinephrine. So without glucocorticoids, if you were having a fight or flight response, essentially you're going to run out of epinephrine in approximately 3 to 5 minutes. The only way to continue that fight or flight response is with your glucocorticoids. And they are going to stimulate the increase in production so that you can have a greater and more sustained response for your fight or flight response. All right? And then finally, we can think about these receptors. So So here's a bunch of here's a standard catecholamine, there's the noradrenaline, here's the adrenaline, and these are the receptors. We have the two different kinds of receptors. We have the alpha receptor, and epinephrine binds to the alpha receptor, and then we have the beta receptors. And epinephrine also binds to the beta receptors. So epinephrine binds to both. The beta receptor is a GS receptor, so it's going to stimulate the production of cyclic AMP. The alpha receptor is a GI. It's an inhibitory receptor. It's going to shut off the production of cyclic AMP. And the norepinephrine binds preferentially to the beta receptor. So most of the effects that we think of are going to be mediated by the beta receptors, not the alpha receptors. The alpha receptors are going to turn things off. So there are going to be different tissues which are going to produce these different receptors. And so the propranolol, your beta receptors, that's why they function. They're turning off this response. Okay? That's how we get catecholamines. Now, what are they doing? All right, so there's a bunch of things that catecholamines do. And remember, these are happening very, very fast. One of the things that they do is they shut off visceral activity. So this is inside the gut. As you're going through a fight or flight response, you actually turn off digestion. That makes a lot of sense. The way I like to think about this, the fight or flight response, is do you want to be lunch or do you want to have lunch? Even if we've already eaten our meal, we can wait to digest it later. We can shut off our digestion. We'll do that later, assuming that we're not dead. Somebody else's lunch. Okay? So we shut that off. Another thing we do is we increase brain blood flow. So we get more blood going to the brain. We vasodilate inside the brain to get ourselves thinking better as we're trying to escape. That will allow us to also increase arousal. Another thing that we do is we increase visual acuity. This is one of my favorite things because it's the increase in visual acuity. We actually see better. The epinephrine allows us to see better. And in some ways, I don't know if anybody had this experience where you're just about to get into a car accident or you turn around, you're just about to smack into the door, and it seems like time starts to slow down. What that is happening is your epinephrine is changing your visual acuity, and the brain can't process that acuity that fast, so it interprets it as time slowing down. Because what essentially, the way I like to think about it, it's not completely accurate, but the way I like to think about it is that all of a sudden your eyes are, to use an analogy from film, it's taking more frames per second. And so, because the acuity is increased. And so by doing that, the brain interprets that as time slowing down. So as you're sitting there turning and you're hitting that door, it's like, oh my goodness, the whole time started to slow down when that was happening. Okay, so we changed the visual acuity. We also actually changed gas exchange. So your efficiency of your lungs increases. You get more oxygen out of the air with epinephrine than without epinephrine. Another one, before we get to cardiovascular activity, we increase piloerection. This may be my favorite word in the entire English language because it sounds so X-rated and yet it's not. Does anybody know what piloerection is? Yeah?
Goosebumps.
That's a part of it. It's the standing of hair up in its follicles. And when you don't have hair, what do you get? Goosebumps. But really, it's the hair standing up. So if any of you have had dogs or something like that and they get upset or anything, the hackles go up, right? That's what that is. It's piloerection. So the erecting of the hairs in the follicles. Yeah.
Why is that beneficial?
Well, one of the things that happens if you get an animal cornered and it's facing a predator, if you can make yourself more fierce, then maybe you can convince that predator to leave you alone. So one of the things you can do is make yourself bigger, at least appear bigger. So the hair goes up. Okay? And one of the more important things that we do is we're changing cardiovascular activity, and we're doing it very fast. We vasoconstrict in order to get blood into the areas where we need it, so especially around the gut, the intestines, the stomach, we're going to vasoconstrict all of those blood vessels. We're going to vasodilate, especially in places like the retina and in your muscles, to get the blood and oxygen and the glucose there faster. And you also dramatically increase heart rate. And the reason I talk about heart rate, I'm going to show you some data on heart rate in a minute, is that that's actually something that can be measured. It's very difficult to measure in real time, vasoconstriction, vasodilation, a lot of these other things. But we can measure heart rate. And one of the reasons, I'm just going to show you some of the data that comes from our lab, but it's true of humans and every other vertebrate that anybody's looked at. This is a transmitter. And you can stick that transmitter inside an animal. In this case, we're doing it in a bird. And that allows you to measure the change in electrical potential across the heart. And so then you can get a trace of the heart rate. And so this is what's happening. This arrow right here is the stressor. And what we can see, maybe I'll kick this up a little bit. What we can see is right after that stressor is applied, a nearly doubling of the heart rate. And I want you to look at this. This is time in seconds. So from 2 to 3 seconds, we've more than doubled our heart rate. This is really, really fast. So anybody who's merely been in an accident or something like that knows that. All of a sudden, your heart is just pounding, and it happens just like that in under a second. And if we look at this, here's basically the picture, by the way, of one of our birds that has this transmitter and is sending that signal to this plate. So this is how we're able to measure this in real time without disturbing the animal. And if you then integrate, so take all of these heart rate traces and just talk about the basal heart rate, and then you apply your stressor, what you can see is this really rapid increase in heart rate that is sustained for about 5 minutes or so until it comes back down. And this is your classic fight or flight response. Now, one of the really fascinating things about heart rate, though, is that the heart rate is regulated from a balance of the sympathetic and the parasympathetic nervous system. So the sympathetic nervous system increases heart rate, where the parasympathetic, so this is the parasympathetic, through the vagus nerve, inhibits the heart rate. So there's 2 ways that you can get an increase in heart rate. If you need to jog for the bus, thank you. If you need to jog for the bus, you increase your heart rate in order to sustain that jogging. It's not actually a sympathetic nervous system response. It is an inhibition of the vagal response. So in other words, you're taking off the brake. You're not hitting the gas. And so most moderate exercise is regulated by a decrease in the negative input. So a decrease in the vagal input, not the increase in the sympathetic. Now the interesting thing about that is that the vagus nerve also regulates our breathing. And so the breathing expands the lungs and sort of puts pressure on the heart. And so in order to sort of fit the breathing rate in with the heart rate, the vagus nerve regulates the interbeat frequency of the heart. So if we look at this as a trace from a normal person and its heart, each of these is a beat, heartbeat. But what you find is that this distance between this beat and this beat is quite different. It's much longer here. And this is what's called heart rate variability, or HRV. Now, this is what happens when the parasympathetic is regulating this process. When the sympathetic regulates this process and epinephrine is flooding into the heart, it becomes much more regular. And so the regularity between heartbeats is then a decrease in the heart rate variability and the variability between beats. And so you can diagnose when somebody is having a fight or flight response with their increase in their heart rate by whether or not you see a decrease in the HRV or not. Why is this important? Okay? It's turning out, as we learn more and more about cardiology and how the heart works, is that a decrease in the vagal input, so decrease in parasympathetic in order to increase heart rate, is pretty benign. An increase in heart rate due to the increase in epinephrine through the sympathetic nervous system is not so benign. If we do that for a little bit, that's fine. But if we have somebody who's under a lot of stress that continues to stimulate their heart through the sympathetic nervous system, we see a long-term decrease in heart rate variability. And that turns out to be a marker for incipient heart disease. So you can start to get a lot of cardiac problems if you have too much activity of the sympathetic nervous system and you see this long-term decrease in HRV. Yeah?
Could you explain how controlling breathing helps maintaining a constant HRV?
It doesn't. That's how you get the variability. Okay? You have to fit that heartbeat in around the breathing. And so what they're doing is instead of going bump, 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 it goes bump, 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 bump. So I'm exaggerating it, but you see a lot of variability between beats in your heart.
Because your breathing goes faster?
No, it's just because you're breathing. You don't want to pump at the same time that you've just taken a big breath. Okay? Because there's so much pressure on the inside of your body.
So this is a sympathetic, a parasympathetic?
That's a parasympathetic regulation.
So this is like normal? This is
Yep.
good.
That's normal and good. Yeah.
Why does decreased HRV help contribute to heart disease?
Because it turns out that it's an indication that the heart is being continually stimulated by epinephrine.
So does it,
And
the
that's
cause
not
is
good.
Just a
It just seems to be a signature of a heart that's starting to get into trouble.
okay.
Yeah.
At what point does heart rate variability is considered arrhythmia or any sort of dysregulation of the heart? Like, how much does it vary in terms of regular heart?
Yeah, so the heart rate variability is defined over a long period of time, 5-10 minutes. So the arrhythmias that people are talking about, or when somebody goes into cardiac arrest and it becomes really arrhythmic, that's really an acute thing. So heart rate variability doesn't really fall into that. Yeah.
Does the parasympathetic signal cause the variability, or if you have lack of both signals, do you have variability?
You have variability when the parasympathetic is primarily regulating the heart. You have no heart rate variability, or very little. It becomes more like a metronome, very, very simple, when the sympathetic is regulating heart. Yeah?
Is there ever a point where there's like neither regulating, like you're not stressed out or, exercising or something else that would cause the parasympathetic nervous system to have an effect? So it's just like...
Well, the parasympathetic isn't working, you're dead. Okay? But the heart does actually beat on its own. So it has its own endogenous rhythm that's done by the cells. So what the vagus is doing really for the heart rate is helping regulate that process. So it's slowing things down and getting it synchronized with your breathing. Yeah.
So you said that the parasympathetic responds to moderate exercise. What about long, 45 minutes of running, right?
Yeah, if it's moderate,
that's considered.
it's still going to be. It's regulated by the withdrawal of the parasympathetic.
And so if you maintain a normal breathing rhythm, during the course of that exercise, would that lead to less heart rate variability?
No, because of the way in which it's being run. Because it's really this process here that creates the heart to beat in such a steady rhythm. Okay? It's this process here that allows it to continue with the variability and to regulate it along with the breathing. Yeah.
So, obviously you can regulate your own breathing rate at times. That's controlled by the parasympathetic then?
Mm-hmm.
So us, like, if we like are hyperventilating, then the reason why your heart rate changes is because your parasympathetic comes into play, or is that when your sympathetic then starts?
No, that's when your sympathetic
it
starts kicking in.
Kicking in.
Yep. If
So
we're
normal
hyperventilating.
everyday breathing, and you like saying like doing like yoga or something like that, that would be parasympathetic and changing that. But if you start hyperventilating and you're increasing your lung capacity and doing all that stuff, then your sympathetic comes into play.
Correct.
Got it.
Yeah.
Could you explain the connection between HRV and heart rate? Because I don't. So this is like the variability in heart rate, but.
Yeah, that's a good question. This doesn't have anything to do with heart rate. Okay? This is just the HRV is the distance between the beats. So, and it's basically a ratio of how much variability there is. So you can get the same kind of variability even when the heart rate increases. That's the beats per minute. This is the interbeat interval. Okay? All right. The other thing that has become very clear is we used to think about the fight or flight response as being an all or nothing response. Then once it's turned on, you get the response, and then you turn it off. So it's binary. It's either on or it's off. Turns out that's not entirely clear, and not true. So in this case, I want to explain this experiment to you, because this is one the experiments one of my graduate students did a number of years ago, and I just love this experiment. Because what we did is, here's the bird that has this transmitter in it. So this is one that we're measuring heart rate in. And then we put 1, 3, or 5 intruders inside a lower cage. They do not like being underneath another bird. This is really, really stressful for these guys. But we weren't measuring them. But there's a trap door between the lower one and the upper one. And so he rigged up this system with a bunch of pulleys and strings where he could open this trap door from outside the room without disturbing the birds. And when he opened that trap door, all of these birds down here flew up into this cage and really started to crowd the bird that we were measuring. So we were looking at the response to crowding. And it turns out, here's the response when there was one intruder that came out, an increase in heart rate. We had a much larger increase when there were 3 intruders, and an even larger increase when there was 5 intruders. So in other words, we can actually titrate the amount of a fight or flight response that we have. It's not an on or off. It's not binary. All right. And finally, just because I think this is one of the coolest experiments ever, somebody talked about freezing earlier when I talked about a fight or flight. So there are some species that really, really freeze when faced with a predator. In fact, they freeze so hard that it looks like they're dead, and they feign death. What do you think happens with their heart rate? Yeah, it plummets. Okay, so here's the data. I love these data. So here's the heart rate of this animal. Here's when it gets faced with a predator. His heart rate just drops, and it looks like it's almost dead. And then the predator leaves at point B. And guess what happens when the predator leaves? Now we have our classic fight or flight response. Massive increase in heart rate. Interesting though, at this point where it has this massive heart rate increase, the predator's left. The predator's gone. So there's no particular need for it at this point. And yet it still has a stress response. It still has an increase in heart rate. Yeah?
I was gonna ask why that is.
Yeah. So we still don't know why that occurs. But a lot of species will do this. If they when you go into that freezing mode, you actually shut down the heart rate and you decrease the amount of epinephrine. But normally what happens is that once that ends, you see this big rebound. and everything starts to happen again. Yeah.
Is that rebound necessary to compensate for the fact that things were shut down? Because like maybe there's not enough blood flow, so now you need to like overactivate blood flow?
Fantastic question. We have no idea. There's so few people doing this work, which I can't understand. I think working with possums and doing this would just be really cool. But a lot of species do this. I remember back when I was a kid, we took our big German Shepherd over to our friend's house. And unbeknownst to us, they had left their pet rabbit out in the backyard. And we put our German Shepherd in the backyard, and the shepherd saw that rabbit, and there was a big, massive chase, and our neighbors were crying because the dog caught the rabbit and killed the rabbit. And the rabbit was sitting there absolutely dead, stone dead, and our neighbors were so upset and crying. And my mother said, well, you know, I don't see any blood on this rabbit. I don't see where the dog actually bit it. It's just sort of slobbering all over it. So we took the rabbit, put it in its hutch, walked away, and 3 or 4 minutes later, the rabbit jumped back up and went over and had some food. Okay? Totally bamboozled our German Shepherd. Shepherd had absolutely no idea what it was looking at. It was probably surprised the hell out of itself by actually catching it. So, yeah, it can be an extremely effective way of preventing being eaten. This is also feigned death. Yeah?
But if the animal's faking death, why doesn't the predator just eat it then?
Yeah, well, our German Shepherd is very well fed, right? So this is probably an interesting case in which it probably didn't need to eat. So why doesn't it kill it? That's probably the only good reason to think about it. But one of the things that's interesting about predators is that, and one of the things that you have to think about if you're a prey. So fight or flight or freeze. So you're sitting there and you're an animal, and you see a predator, and the predator's coming, but it hasn't detected you yet. You have one of two choices. You can run, but one of the things we know about running is that you are going to guarantee that you're going to be chased, because that's so ingrained in a predator. It sees prey running, boom, go after it. Or you freeze and hope that the predator didn't see you. The problem with, and so you might not have to run, you're not going to be chased. The problem then is that you're going to let that predator get potentially lethally close if it actually has detected you. So you're going to have to make that trade-off. And so what probably happened in this case is that you had a predator that really actually wasn't that hungry, but as soon as the rabbit ran, it guaranteed a chase. Yeah.
Is that like when people end up like running into bears in the woods and stuff, and they freeze automatically?
Yeah. You're generally much better, because if you start to run, you're basically guaranteed a chase. Yeah.
Well, from a like predator perspective as well, if you're coming across something that looks dead that you didn't kill, you don't know why it died, so you have issues with like disease or things like that, so there might be a natural avoidance system for that to try and avoid disease later on.
True, but I'm not sure why the German Shepherd didn't think it just killed that rabbit.
Yeah.
So, it's a pet. It had never killed anything like that. It certainly didn't have to, hunt its own food. So, all of a sudden this rabbit just lying there that it was chasing, I'm not sure why it would think that it didn't kill it. All right. All right, so that's the end of fight or flight. Where's my... There. So that's this process. Now let's go over to this slow response. So we have the adrenal cortex, cortisol, everything, all of these things that the glucocorticoids are doing, and we're going to go over these for the rest of today and next week. So this is mostly a review, I hope, in what is called the HPA axis. The hypothalamic pituitary adrenal axis. So we have a stressor. It sends a signal to the hypothalamus where we get the release of CRH and arginine vasopressin, or if it's a mammal, arginine vasopressin, which then travels through the portal system to the pituitary where we get the release of ACTH ACTH impacts on the adrenal cortex, and where you get the release of glucocorticoids. The primary glucocorticoid in mammals, most mammals, and fish is cortisol. The primary glucocorticoid in reptiles, birds, amphibians, and for some reason that nobody has ever given me a good explanation for, rodents, they secrete corticosterol. Okay, so the two main, and so there's, it essentially have the same types of effects on the body. They then travel through the blood with CBG, which is corticosteroid binding globulin. So this is our carrier protein that carries the steroids to their target tissues. We then have negative feedback. Negative feedback exists basically at two levels. One at the level of the hypothalamus, but the more important one, and we talked about this before the last exam, is the hippocampus. And so negative feedback stimulates the hippocampus, which sends a negative signal to the hypothalamus as well. So we get two different feedback loops. Hopefully that was all reviewed. Okay? What we're going to talk about now is what all of these effects are when they impact with their receptors. But thinking about this whole HPA axis, and getting to the point where you get the release of glucocorticoids actually takes a little time. And so this is the type of response that we get to see. So this is a figure I made many years ago talking about capture of wild animals, but you can just say time after stressor. And so here are your glucocorticoid levels. And what we see is that it takes approximately 3 to 5 minutes from when the stressor is detected by the animal before it starts increasing its glucocorticoid levels. So in other words, if you had measured the glucocorticoid levels prior to that stressor, you would have seen something about like that. So they're very, very stable, and they don't start to increase because you have to get that entire cascade through the brain and then get the ACTH all the way down to the adrenals, and then you start to see the increase in your glucocorticoids. And then, if you monitor those change over time, what you see is a major increase in your glucocorticoid levels. They peak at usually somewhere between about 30 minutes. And if it's not a sustained stressor, what you'll then see is a decrease through negative feedback. And this will, somewhere between 2 and 3 hours, will come back down to its original levels. Yeah.
So if the stressor is only like instantly present, would the levels still increase and then drop only after 30 minutes?
In general, no. In general, you need to have a stressor sustained for 5-6 minutes or so before you get this full response. So if it's just something that takes place for 10 seconds or something like that, then you don't. Okay, so once the glucocorticoids are released, they go to their target tissues and they have their effects in the cell. This is the same figure I showed you at earlier in the semester with the steroid receptors. So it's not any different than what I showed you before. It's just repeating. Is that the glucocorticoid receptor is in the cytoplasm. It binds to its target, the hormone. It then translocates into the nucleus, dimerizes, and starts the production of new protein. And those proteins then get translated into the mRNA. The mRNA gets translated into proteins, and you get new biological effect from those proteins. So again, this is probably all taking about 30 minutes. So not only are we seeing an increase, it's taking about 30 minutes to see the peak in glucocorticoids, we're also seeing about 30 more minutes before we start to see a major effect. Start to make sense? Okay, so major stress hormone, epinephrine and cortisol. A lion jumps out to eat you. Is cortisol going to be important? Not to get away from the lion. So why in the world are glucocorticoids being released? I'll let you think about that one because the answer will come next week. All right, so those are the receptors. But there are two types of receptors for glucocorticoids. We talked about this a little bit last week. We have the type 1, or the MR, which is really supposed to be binding aldosterone, and type 2, which is the GR, the glucocorticoid receptor. Now, if we look at affinity, so which receptor do the glucocorticoids want to bind to, then MR is much bigger than GR. So as your glucocorticoids start to rise, they want to bind. So this is the physiological behavior, the behavioral response to the amount of binding. The glucocorticoids are going to bind to the MR first. But the capacity, so essentially the number of receptors, GR is much greater than MR. So we saturate the MR very quickly, really at the circadian baseline level. And then we start to bind to the GR, and it starts to increase. Okay? So we bind to the MR first, and then the GR. So what we think is that the MR is involved in sort of basal functions. Circadian rhythm, glucose regulation, etc. Sorry. So this is really the basal functions where these, the GR, are mediating the stress functions. There's a lot of MR in the brain that we do not think is being bound to aldosterone, but is being bound by glucocorticoids and affecting a lot of the basal functions. Okay? All right. So, I'm going to come back to this. We have the stressor. And the stressor goes through what I like to call a discrimination or determination function in the brain before you can rely upon survival. What do I mean about that discrimination determination function? This is where we come back to what I talked about at the beginning of the class. What is a stressor? Well, the brain has to figure out whether something is a stressor or not. And one of the big things that we do is, in thinking about this, is in terms of what can cause an effect and what isn't. But a lot of it can be dependent upon the context. So let me give you an example. I want to show you a number of really classic studies that were basically done back in the 70s. So there are graded responses to the amount of glucocorticoids that are released. So here's the experiment. So this is our initial levels of glucocorticoids. No, this was all done in rats. So if you walk up to the cage and you pick up the rat and handle it for a little while and put it back into the cage, it has a little bit of a stress response. It has that much. If you then take it out and put it in a new cage, now all of a sudden it has a much bigger response. If you take it out and put it in a cage that doesn't have any bedding in it, it has an even larger response. And if you take it out and put it in a trash bucket, empty, it has an even greater response. Why? What is it different about each of those things that makes it different? And the answer is novelty. So the more novel a stimulus is, the greater the glucocorticoid response. Here is another, this is one of my favorite examples ever of this. In terms of the context makes a huge difference. So let me explain this experiment. So there were a bunch of new recruits for the Norwegian Army, and they were going to be trained to be paratroopers, jumping out of airplanes with parachutes. So what they did was they strapped the parachute onto their back and took them to the top of the tower and pushed them off. Guess what happened? They had a massive stress response. They just got thrown off a tower. So here was their original levels, and here are the levels right after they got tossed off that tower. Not too surprising, right? But guess what? They didn't die. So when they took them up the next day and threw them off, then the next day and threw them off, by the third day, here's day 3, their responses were almost back down to normal, and by certainly 5 days, they were completely normal. Same exact stimulus. They were still being thrown off that tower, and yet they were no longer having a stress response. Why? It was no longer novel. They knew this was going to happen. They knew it was they were going to survive. No problem. Yeah.
Would their stress levels return to how they were initially if inside of a tower they were jumping from a plane?
Great question. They never did that experiment, but I would have loved to have done it, and I predict what they would have seen is this. That when they took them up into that airplane and threw them out of the airplane the first time, they would have had a massive stress response. And then my guess is by 3 days later, they wouldn't. They would have learned that even going out of an airplane is not going to kill them and is no longer novel. And I think this explains why sometimes, if you took me up into an airplane right now and threw me out, even with a parachute, I'd have a massive stress response. But a lot of people do this for fun, right? It's skydiving. And I think the difference is that once you learn, it's no longer novel. And so novelty is a major, major component of what causes stress and what doesn't. Yeah.
If they saw like someone else getting hurt or dying from it, do you think the stress would go up afterwards?
So in general, people have seen that, but not as much as you might think, because they knew that they were okay. Yeah?
If they stop doing that for a long time and then do it again, would the stress level change?
It depends. Sometimes yes, sometimes no. It depends upon how novel it was and how maybe how stressful it was at the beginning. But this will start to explain to you why, if you walk into an exam and you didn't study the night before, you have a much bigger stress response than if you did. Because if you'd studied, it's no longer as novel. You know what to expect. Yeah.
So by novel, I mean like unexpected?
Or just new. Maybe unexpected, but it's mostly new. You can expect it. Like I'm giving this major lecture at my meeting in January. I know it's coming. It's not unexpected. But I've never given a lecture in front of 400 people before. I'm already starting to get stressed. Okay? So it's novel. I've never done that before, but it's not unexpected. It's really the novelty. All right? All right. Let's stop there and we'll pick it up. Have a great Thanksgiving.
I have a little more